Is my screen is visible? Just let me know. Then we'll proceed further.
Yeah, sir, it's visible.
Okay. Kanji. So the topic for today's session is DNA tools, gene cloning, DNA tools, gene cloning 2, DNA tools and biotechnology, DNA tools and biotechnology 2. So my name is Ankit Kumar Malik, an NPTEL PMR from IIT BHU. So the course instructor for the same course is Professor Sanjeeva Srivastava from IIT Bombay. So let's proceed further from the question 1 of the last year assignment. Am I audible to all?
Yeah,
Okay. So
audible.
question 1 is Anisha wanted to perform a PCR experiment to amplify her gene of interest. So she chose the following two primers, like forwarded primer and reverse primers are given here. So she followed all the steps, but still do not get any product. But could be the possible? What could be the possible reasons for this? So the option A is primer are too short. The option B is primers have high GC content. Option C is the primers are complementary and will form dimers. And the option D is the primers are too long but have provided a high melting temperature. So anybody would like to highlight this question or like answer this question with proper reasons and clarifications? Anyone? Take your time.
So mostly, I think it. So mostly, I think it might be B.
It might be what?
B.
Why? Any reasons?
Mm, Sir, exactly, I don't know. Like
Okay.
mostly, I'm feeling it's B.
Okay. So moving before moving forward, let me explain and all the details and basic concept about primers and the PCR experiment. Then we'll come to this question and we'll rectify all the doubts of whether it is option A, B, C, or D. Okay. So you're confused.
Yes,
You
sir.
know the answers primers having high GC content must be the reason, but you don't know the reason. What actually, why actually it is. So let's start from the basic concept. Then we'll know the reason. So what primers? What is primers? Primer is a short single-stranded nucleic acid sequence that serves as a starting point for the synthesis of a complement strand of DNA or RNA. Primers are essential components in techniques such as PCR, DNA sequencing, and various molecular cloning methods. So we have here PCR process workflow, qPCR work process flow, and endpoint PCR process. So here are some ideal properties of a primer like specificity, length, melting point, GC content, avoiding self-complementary, avoiding homopolymer runs, three-end stability, absence of palindromic sequences, avoiding primer dimers. So we'll go through one by one from specificity. What specificity is? Actually, primer should be designed to bind specifically to a target, any DNA or any RNA sequence. So this ensures that the amplification or synthesis occurs only at the intended region. So this is what specificity do works. Then the second property is length. The length of a primer is a crucial. Typically, primers used in PCR are around 18 to 25 nucleotides long. So this length provides sufficient specificity while allowing efficient hybridization to the target sequence. Then the third property is melting point of any primer. So the melting point represents the temperature at which half of the primer binds to the template DNA. It is important to choose primers with similar melting point values to ensure optimal annealing during the amplification process. And the next property is GC content, which you are having doubts in. So what GC content? The percentage of guanine and cytosine bases in the primer sequence affect its stability. So the balanced GC content, which must be around 40 to 60%, contributes to stable primer template binding. Then the next property is avoiding cell complement. So it primer should not have significant regions of self complementary, as this can lead to the formation of secondary structures affecting primers, annealing and amplification efficiencies. And the next property is avoiding homopolymer runs. So the long stretches of the same nucleotide should be avoided in primer design, as they can lead to mispriming or non-specific amplifications. And the next is 3-end stability, instability of the primers. The third end of the primer is critical for proper binding and initiation of DNA synthesis by polymerase. There is an enzyme called polymerase. This is the main reason for the DNA synthesis, so which ensures the 3-end is stable and does not contain any mismatches with the template. And the next and the important property is avoiding homopolymer runs. Basically, many researchers, many scientists do this kind of mistake, which they do not avoid homopolymer runs. So what homopolymer runs is long stretches of same nucleotide should be avoided in primer design, as they misleading to the mispriming or non-specific amplifications. So the next is absence of palindromic sequences. So primer should not contain any palindromic sequences, as they can form hairpin structures or primer dimers interfering with PCR amplifications. And the last and the most important property of a primer is avoiding primer dimers. This is also the uh, most general mistakes which scientists or researchers do during the primer synthesis or designing of a primer is primer dimers. Primer dimers is formed and amplified in 3 steps. First, I'll explain what primer dimers is. It can occur when primer bind to each other rather than to the template. So, designing primer to minimize and the likelihood of forming primer dimers. So, by considering all these properties during primer design, researchers can enhance the specificity and efficiency of DNA amplification or synthesis reactions in various molecular biology applications. So, the next is primer dimer formation. So here the answer could be GC content also, and the complementary and primer dimers also. So we'll go in brief.
Yes, sir, got it.
Got it now? So let's discuss about primer dimer formation. So primer dimer is formed and amplified in 3 steps. And in the first step, two primers annealing at their respective 3 ends. If this construct is stable enough, the DNA polymerase would bind and extend the primers according to the complementary sequences. Then an important factor contributing to the stability of the construct in step is step 1, with the high GC content and the 3 end and length of the overlap. And the third step occurs in the next cycle, when a single strand of the product of step 2 is used in the template, to which fresh primers annealing, leading to the synthesis of more primer dimer formation. So here the answer will be the primer are complementary and will form dimers. The third one is answer. Let me wait. So is my this animations is working? Just let me know. Is my
No,
animation
sir,
is
only
working?
the question one screen is visible.
So screen is not changing, na? Slides are not changing.
Yes, sir.
No, sir. Slide is not changing.
So now is it changing?
Yes, sir, now it's changing.
Okay, so there is some problem in like slide changing in slideshow. Okay, just give me 2 minutes. Let me rectify this. Actually, I have inserted some animations without slideshow. Just give me 2 minutes. Now, hello, हाँ जी अभी.
Yes, sir.
Yes, sir. Visible.
Okay, so moving to the next question. हाँ जी, does anyone have any doubt? Okay, so let's proceed to the question 2. Question 2 states that plasmid has multiple restriction sites. However, some of them were blocked due to specific modifications on them. Like digestion using ADE resulted into bands of 6 kb, 8 kb, 1.5 kb. And the second is the digestion using BEA resulted into bands of 8 kb and 7.5 kb. And the third digestion, which is by using BC and E, resulted into bands of only 15 kb. So based on the above given information about plasmid map and gel pattern, which of the following sites would be blocked? So anyone would like to answer this question with some specific reasons, with some specific explanation? Anyone? Anyone? Okay, so no one. Okay, so let me start the explanation of this. If anybody would like to take some time or would like to explain this, they can. Okay. So here, uh, when we cut from ADE, which results into 6 kb, 8 kb, and 1.5 kb. So while digestion using BEA, so we are getting 8.8 kb and 7.5 kb band, which means restriction enzyme A and E are working finely. So by using the BEA, so that A and E are working finely because we are getting two bands of 8 kb and 7.5 kb. And while B is not working properly, which A and E are working properly. So by using the third digestion method, while cutting from B, C and E, so only we are getting one band which is of 15 kb. So from our digestion, we got to know that the A is working finely, and from here we got to know that it is cutting at just at one point. Here we can say that B and C are blocked. So during this restrictions, so B and E are not B and E are blocked. B and E sites are blocked. So moving to the basic concept for this are the principle of restriction enzyme digestion principle. So here to analyze the genomic DNA, we must first cut into small must first cut into smaller pieces from manageable pieces. The tools for this restriction enzyme recognizes specific nucleotide pair sequence in DNA called a restriction site and cleaves the DNA, which basically hydrolyzes the phosphodiester backbones within or near to that sequence. So all the restriction enzymes cut DNA between the carbon and the phosphate moiety of the phosphodiester bond, so that fragments produced by the carbon and the phosphate moiety by the phosphodiester bond may restriction and enzyme digestion may have phosphates and hydroxyls, and the most restrictions enzyme functions optimally at the restriction enzymes are used to produce a pool of DNA fragments to be cloned. So the most commonly used restriction enzymes recognize four nucleotides pair. So here I have mentioned some example of the restriction enzymes. So which are HHA1, ECO R1, BAM H1, NOT1, Hinfongs at BXT1. So let me explain all of these. So this restriction enzymes recognize four nucleotide pairs. For example, HH1 or six nucleotide pairs are called BAMH1 and EcoR1. And some enzymes recognize eight nucleotide pair sequences. For example, NOT1. And other classes of enzymes do not fit our model because five nucleotide pair base sequence in which there is symmetry in two nucleotide base pair or either side by the central nucleotide pair. But the central nucleotide pair is obviously asymmetrical within the sequence, which is called BEXT1, which is the representative of a number of restriction enzymes with a non-specific spacer region between symmetrical and asymmetrical sequences. So the next question is Ramesh has been provided with a map of a plasmid of P3000 whose size is 15 kb. Ramesh observed that the following fragments with different combinations as mentioned below, that is not one with EcoR1 gave two bands of 4 kb and 11 kb, and the second combination is BAMH1 and STU1 gave two bands of 9 kb and 6 kb, and EcoR1 and STU1 gave 6 kb and 9 kb two bands. So upon cutting the plasmid with BAMH1 and EcoR1, which of the following fragments would Ramesh observe? Whether it is 2 kb or 13 kb, or 3 kb and 3 kb and 12 kb, 4 kb and 11 kb, or the last option is 5 kb or 10 kb. So anyone would like to answer this question with some proper clarification? Anyone? Okay, so yesterday also we had session na, on this. Yes or no?
So we have
Um,
today only.
Sorry.
so yesterday for a long time nobody came actually in that session.
Okay. So how many of you was present in the last yesterday's session?
Sir, I was present. Then
Okay.
for a long time nobody came, and like the teacher was also silent. So like just left.
Okay, let me ask someone this question, and then at last I'll explain the explanation for this. So like this, Rajdeep Roy. Rajdeep Roy.
Yes, sir.
So could you please highlight this question and the explanation also? Please.
So I wasn't present in last class.
You were what?
So I was not present in the
He
last
was not
class.
there in the last class. Okay, so let me ask someone else. Uh, Kashish Thakur. Kashish. is not responding. Maybe mic is not working or speaker is not working. So Krishna Chakravarti. Krishna Chakravarti,
Yes, sir.
kindly explain this question to all of us.
Sir, I'm not present in last class.
Okay, I think most of you guys are not present in last class. Okay, no worries. Let me start the answer or explanation of this doubt. So here we have three combinations of not 1, eco R1, BAM H1, STU1, eco R1 and STU1. So upon the cutting the plasmid with the BAM H1 and eco R1, which of the following fragments would Ramesh observe? So here, to determine the fragments obtained by cutting the plasmid with BAM H1 and eco R1, we need to analyze the information provided the fragments resulting from different enzyme combination. So let's find the fragments obtained by cutting the plasmid with BAM H1 and eco R1. So this is the explanation. So we have three combinations. First we have this not 1 eco R1 gave 4 kb and 11 kb. So here if we'll see not 1 to eco R1. So this region is of 4 kb. Hmm? Then BAM H1 sorry 4 kb. This one is from not 1 to eco R1, and this way it is coming 4 kb, and in this way it's coming 11. Okay, 3, 6, 9 and 2, 11. So it's giving 4 and 11 kb. Then second combination is BAM H1 and STU1. So where is BAM H1 and STU1? So this is BAM H1, this is STU1. So from here it's coming 6 kb in this way, and on another opposite way it's coming 9 kb, like 4, 5, 6, 7 and 8, 9. So we have two pairs, one of 6 kb and one is of 9 kb. And the third combination is eco R1 and STU1, which gives 6 kb and 9 kb as well. So let's find out eco R1 and STU1. So where is eco R1? Here it is eco R1 and STU1 here. So let us count from in this way. It's coming 4 plus 2, totally 6 kb. And in another way, it's coming 6 plus 3, 9 kb. So this the combination are justified. So now we have to calculate the plasmid by cutting the plasmid with BAM H1 and eco R1. So if we will, where is BAM H1 and eco R1? Okay. So in this way, it's coming 3 kb, and in this way, it's coming 6, 7, 8,
12.
and 4, 12. So what could be the possible combination it could be?
So 12 and 3.
Exactly. 3 and 12. Got it, na? You got my point, na?
Yeah, sir, got it.
BAM H1 and eco R1. From this way, it's coming 3, eco R1 to BAM H1, and BAM H1 to eco R1 is coming 12. So the best answer for this is 3 kb and 12 kb, that is P. Okay.
Yes, sir.
Okay. So moving forward to the next question. Question 4. So here you have been given a plasmid of 9 kb. You have to perform a cloning experiment for which the target gene is GFP, which is green fluorescent protein. So given below is a schematic representation of the plasmid and the target gene. The green fluorescent protein is of 0.7 kb, which is successfully inserted in the plasmid. So choose the correct statement. So the size of the plasmid after cloning green fluorescent protein in it would be 8.8 kb, or the size of the plasmid after cloning GFP in it would be 8.2 kb. I think both are same. I've copied
Yes,
both
both are
options are same, na?
same.
Both the options are same.
Sir, both are same.
Actually, it's a copy and paste mistake. It's mine. Sorry, no. It's 8.8 kb and 8.2 kb. Both are different, man.
Acha, okay, sir. Yeah.
Even I got confused. So the option C is The size of the plasmid cannot be determined with the data provided. And the option D is the size of the plasmid after cloning green fluorescent protein, and it would be 9.7 kb. So, would you like to explain this, man? Anyone, any can could come and explain this. Actually, this is a not an proper teaching session. This is a discussion session, problem solving session. So you can also come and discuss any question, like your all the mistakes, your all the doubts have been cleared here. So if you have any like little bit knowledge, you can come outside and just discuss this. Anyone could. So I'll wait for
Sir,
1 minute.
maybe option B.
Option B. And why option B, man?
Sir, because actually, to insert this into the target gene, you need to cut it actually.
Okay.
So after cutting it and again inserting it, almost
And
like
what?
8.3.
Okay, in what way we will cut? Like we have to cut, but there is some procedure. There is some specific region to cut. So which region we have to replace, or which region we have to clone, or at what sites we have to cut?
Sir, from this eco-RI to not one region, we'll have to cut and we'll have to put the gene there.
Okay, so let me explain this. You are right. So according to that 8 point, the option B is the correct one. Okay.
Yes, sir.
Here is the explanation for the same. So we have the given plasmid of 9 kb. Hmm? So in this given plasmid,
Yes, sir.
from eco R1 to not 1, it is coming 1.5 kb, and not 1 to stu 1 is coming 3 kb, and from stu 1 to eco R1 is coming 4.5 kb. Hmm. Here also it's missing kb. So this is the given information by the professor or the anyone. So after cloning experiment, so after adding this 4.5, 3 and 1.5, it's coming 9 kb. Okay. And after
Yes,
cloning the
sir.
experiment, so we do not have to do anything in this. We just have to replace this 1.5 kb with this 0.7 kb. So after replacing this 1.5 kb eco R1 to not 1 by 0.7 kb eco R1 to not 1. So after replacing this 1.5 kb from by 0.7 kb, it's coming 8.2 kb. So got it?
Yes, sir. Got it.
So anyone else have any doubt? So they can ask. This is just a simple thing. This is normal calculations. Does anyone have any doubt? They can ask. If they are shy to open up their mic, they can drop the question in the chat box also. Okay, so let me clear the basic concept of gene cloning in this experiment. What we did in the experimental things. So here we did gene cloning. There is one term called gene cloning. So we'll cover the basic concept and basic principles of the gene cloning. What gene cloning is, and what steps comes in occurs in the gene cloning procedure or experiment. So here it is the gene cloning. So certainly gene cloning involves the production of identical copies of a gene or a DNA fragment. So here are the key steps in gene cloning, which is summarized as there are some different steps called isolation of DNA, then selection of a vector, digestion with restriction enzymes, then ligation, transformation, selection of transformants, screening and identifications, expression of cloned gene, harvesting and purification. So we'll go through one by one. So the first step is isolation of DNA. So the first step is to isolate the DNA containing the gene of interest. So this can be the genomic DNA or any complementary DNA or cDNA synthesized from messenger RNA or mRNA. So moving forward to the next step, which is a selection of a vector. Vector is a carrier DNA molecule into which the target gene is inserted. Common vectors include plasmid or any viral vectors. So the choice of vector depends on factors such as the size of gene and the host organism. So the next thing is the digestion with the restriction enzymes. So both the vector and the target DNA are cut using restrictions enzymes. These enzymes recognize specific DNA sequences and there they create these sticky ends that facilitate the subsequent joining of the vector and the gene of interest. So the next step is ligation. Whatever the gene of interest is then ligated to the vector using DNA ligase. So this result in a recombinant DNA molecule where the target gene is now incorporated into the vector. So leading to the transformation. So the recombinant DNA is introduced into a host organism, usually a bacterial cell, through a process called transformation. So this can be achieved through heat shock or electroporation. So the next thing is screening and identification. So PCR or polymerase chain reaction or other screening methods are used to confirm the presence of the gene of interest in the transformed cell. And the last step is harvesting and purification. So here, once the gene or protein of the interest has been successfully cloned and expressed, it can be harvested and purified for further analysis or further characterization or use. So moving forward to the next question, which is question 5. So uh, here some given statements are mentioned. So the statement 1 is lactose bind to the RNA polymerase and increases efficiency. And the statement 2 is the lactose binds to a repressor protein and alters its conformation to prevent it from binding to the DNA. And the statement 3 states that the lactose binds to an activator protein which can help the RNA polymerase binds to the promoter and being transcription. And the statement 4 states that the lactose prevents premature termination of transcription by directly binding to and deforming the DNA. So the question is which of the following statement is correct regarding the activity of lactose in promoting transcription of LAC-Z? So the options are the statement 2, 1, 3 or 4. So anyone would like to answer this question with some reasons? Anyone?
Sir, option A.
Okay.
Sir, what happens is in lacoperon, when lactose binds to the repressor protein, then what happens is like the polymerase, then the lactose can easily go through the um, go through the go metabol go through its digestion actually.
Mm-hmm. Okay.
Then it'll be easier for it. So the repressor will like um, stop the polymerase from
Okay. Exactly.
going and activating the factors for digestion
Correct. Correct.
of lactose.
Correct. Statement second is the correct statement regarding the activity of lactose in promoting the transcription of lactate. So, which states that the lactose binds to a repressor protein and alters its conformation to prevent it from binding to the DNA. So, here uh, there is one clarification and basic concept of lag operon. So, do you guys know what is lag operon is? Anyone would like to highlight lag operon? Basic definition of lag operon. Anyone? A simple definition.
Mm, so lactoperon is basically used
Mm-hmm. Go ahead.
basic, basically used for digestion of lactose.
Okay. So exactly. So let me start the proper clarification and concept behind these statements. So the lag operon is a classic example of an inducible operon in prokaryotic organisms, specifically in bacteria like E. coli, which plays a crucial role in the regulation of lactose metabolism. Let's break down the concept of the lag operon by using the absence of the inducer and by in presence of the inducers. So the let me explain the structure of lag operon. The lag operon consists of three main components. the structural gene, the promoter, and the operator. Then the control of the repressor protein. So what a repressor is? So the lag operon is usually turned off, which is called repressed when lactose is absent. So this repression is controlled by a regulatory protein called the lag repressor. So then induction of lactose, I told you the absence and presence of the inducer. So the induction of lactose, when lactose is present, it acts as an inducer. Whereas the lactose binds to the lag repressor, causes conformational changes that prevents the repressor from binding to the operator. Without the lag repressor bound to the operator, RNA polymerase can initiate transcription of the structural genes, leading to the production of beta galactosidase and lactose permeases. So the option uh, answer A and of statement 2. The lactose binds to repressor protein and alters its conformation to prevent it from binding to the DNA is the correct statement. Moving forward to the next question, which is question 6. So look at the given figure carefully. Real-time PCR was performed with the breast cancer cell 1 and normal 2 genomic DNA samples from the same patient. So what would be the most appropriate reason that fluorescence did not increase after cycle 30? So here as you can see, cycles are given here from 2 to 30, and after 16 there is an hype in the fluorescence, and but after 30 cycle 30 there is no increase in the fluorescence after cycle 30. So what would be the appropriate reason for this? So the possible reasons are like all the primers molecules have been utilized in the reaction, all the template molecules have been used up, or the DNA polymerase becomes limited in the reaction mixture. Anyone would like to answer?
So both the statements A and B actually,
Okay,
like when primer molecules have been used up and template molecules, all primer molecules are utilized and template molecules are used up, then mostly like the binding is done actually. So
okay,
there will be nothing further reaction to go on.
okay,
So it'll just stop. So it just stopped at that point.
okay. So are you sure that option A, both A and B? You know
Yes, sir.
why
Sure.
all the templates molecule have been used up? There are two things, like primer molecules and template molecules. So fluorescence basically works for the primary molecules, not for the template molecules.
Okay, sir.
So according to this, okay, I'll explain the reason also. I'll give the reason also, then we'll back to the answer. Okay. In the next
Yes, sir.
slide, we have some justification for the fluorescent dye based RT-PCR and DNA probe based real time PCR. So you know what real time PCR is? Any definition or any layman explanation regarding RT-PCR? Hmm? Anyone? Okay, let me explain the definition of RT-PCR. So, real-time PCR, as the name suggests, measures the amount of polymerase chain reaction products at the end of each amplification cycle. So, to do this, you label the PCR products, polymerase chain reaction products, during or after the primer extension step of each amplification cycles. And here the fluorescent dye based real-time PCR, here the fluorescence intensity is proportional to the amount of PCR product during the exponential phase of the reaction. And on this relationship rests quantitation in real-time PCR with each amplification cycle, where there is a proportional change, where there is a proportional increase in the fluorescent intensity. Like if you start with more copies of the template which reach the threshold fluorescent intensity, so in fewer amplification cycles, the number of cycles it takes to get this intensity threshold is called to target DNA CT value. There is one thing called CT value. You can use the CT value in one or two ways. So one like you can compare it to a standard curve, which is a semi-log plot, to the CT values versus the log of known concentration for your target DNA. So you then interpolate from this plot to determine the absolute copy number of the target DNA or RNA. So the second one is to co-amplify a housing gene either in the same tube with a multiplex experiment or amplify in a separate experiment. So this is the real-time PCR in a nutshell. So coming back to the question, so here there are two things. One is the template molecule. The second one is primer molecules. So if we used all the template molecules, there will be no such changes in the fluorescence property could be there. And if we use all the primer molecules now, so after 30 might have all the primer molecules have been utilized. That is why there is no increase in the fluorescence occur after cycle 30. So got it, man? So template
Yes, sir,
molecules
got
and
it.
primary molecules. So primary molecules are the primary things for the fluorescence in breast cancer and the genomic DNA samples. Okay, so moving to the next question, which is the seventh one. So the question 7 states that they read the given statements carefully. So the statement first is the agarose gel acts as a molecular sieve which separates nucleic acids based on a molecular weight. And the statement second is the DNA molecules move towards anode. The statement says DNA binding dye is mixed with fluorescence in UV light. And the statement D states that the agarose gel acts as a molecular sieve which separates nucleic acids based on molecular structures. And the last statement states that the DNA molecules move towards cathode. So, which of the following combination of statements is or are correct about agarose gel electrophoresis? So, which student would like to answer this question? Take your time. Take your guess. Take your time. Anything.
Sir, options A, B, and C.
Okay.
Like
Why?
option C.
Why option? Why
Um,
statement A, B, and C?
Sir, what happens is in agarose gel electrophoresis is based on the molecular weight. It'll cut the like nucleic acids into separate fragments, and DNA molecules by their nature they just move towards the anode,
Okay.
and like when the dye is mixed, it shows fluorescence in UV light actually.
Okay. Yeah. Correct. Let me show you the last statement. What have you told now? The UV light fluorescent. Let me show you. So,
Yes,
is
sir.
this thing is visible to you?
Yes, sir, visible.
Yes, sir.
Yeah, exactly. So these are the DNA bands separated on gel. So the length of the DNA fragments is compared to a marker protein fragments of unknown length in UV visible spectrum in a UV chamber. So, am I clear?
Yes,
So
sir.
here two images are there. So the first illustration of the DNA electrophoresis equipment used to separate DNA fragments by size, where a gel sits within a tank of buffer. So the DNA samples are placed in wells at one end of the gel and an electrical current passed across the gel. So the negatively charged DNA move towards the positive electrode. So here this is the proper illustration of all the bands. Okay.
Yes, sir.
So those option C, which is statement A, B, and C are correct. So the agarose gel acts as a molecular sieve which separates the nucleic acid based on their molecular weight. As you can see, according to the molecular weights, all the DNA bands will be get separated. So this marker is a housekeeping marker. Rest all the DNA fragments will be separated or distributed according to their molecular weight. The statement B is DNA molecule move towards anode. And the option C, the statement C is DNA binding dye is mixed, which fluoresces in UV light, as you can see here. So here it fluoresces in UV light. So moving to the next question, this is question 8. So Shruti wanted to perform a PCR reaction. So therefore she decided to design a primer. So she calculated the melting point of the designed primer to be 64 degrees Celsius. So she selected the temperature flow in the thermal cycler as 95 degrees Celsius, 64 degrees Celsius and 72 degrees Celsius for denaturation, annealing and extension respectively. So choose the correct statement from the following. So the statement first is the PCR reaction would fail as the extension temperature needed to be set at 60 degrees Celsius. And the statement B states that the PCR reaction would fail as the denaturation temperature exceeds the melting temperature of the primer and the primer would also denature. And option C states that the PCR reaction would fail as the annealing temperature was needed to be set at 72 degrees Celsius and extension temperature at 64 degrees Celsius. And the option D states that the Malti has not done any mistake and the PCR reaction is most likely going to be successful. So, anyone would like to highlight the statements? Anyone with their expertise? This is a very interesting question, by the way. Anyone could jump in and just explain this.
Sir, option D, all the steps have been followed and everything seems right. The denaturation temperature, annealing, and extension temperature all seem right. So mostly it will go successful.
Okay, so and here there is one statement is there that she calculated the melting temperature of the design primer to be 64 degrees Celsius. So, do you guys know how she calculated the melting temperature of the design primer? How did she calculate it that it is 64?
So by counting the A and the T and G and C, there is a formula like A plus T into 2 plus G plus C into 4, which gives the melting temperature.
Okay, okay, exactly. You are correct, man. The option D is the correct one. So let me explain this. So here, this is the calculation of the melting temperature. Is my screen is visible to all?
Yes, sir, visible.
So do you get anything with this slide? Can you explain this slide? This is
Sir, actually,
saying exactly what you have told now. So just explain this
sir,
slide again.
actually, first we counted the number of G's,
Okay,
then the number of C, then they all added to 12 actually. They
okay,
multiplied it by 4. Then
okay,
we counted the number of A's and number of T's.
okay,
They added up to 8. So we just multiplied them and solved the formula and we got 64.
okay. Exactly correct. So here there is a formula for the calculation of melting temperature of the designed primer. So which is 4 G plus C content plus 2 A and T content. Actually this is the typing error. It will go as the plus. Okay. So
Yes,
like
sir.
in the question states that she calculated the design primer, the melting temperature of the design primer is 64. So I have designed one primer likewise that the probable temperature of the design primer will go 64. So here this is one example is there. So here like G and C content, when we calculate the number of GC content, so it's coming 12, like 1 2 3 4 5 6 7 8 9 10 11 12. Okay, so G to C content is totally 12 and AT content is like 1 2 3 4 5 6 7 and 8. So by applying this formula to this data, so we have like 4 and GC content is 12, then AT content is 8. So by multiplying and adding using board mass method, so it's coming 64 degrees Celsius accurately what the Shruti got the results. So okay, so according to this, the statement D is the correct statement. So the Malti has not done any mistake, and the PSA reaction is most likely to be going to be successful. So moving to the next question, resistant genes. Hmm. So read this given statement carefully. So there are some assertion and reasons are here. So the assertion is a vector has a streptomycin resistant gene of its own. The hint 3 cut site is present in the penicillin resistant gene. If hint 3 restriction digestion was performed to insert the gene of interest, the bacteria which did not survive in penicillin mixed plate but survived in streptomycin added plate are the required transformants. Though, so the reason for this assertion is the unsuccessful transformants would have made the bacterial cells sensitive to streptomycin. And the possible options for this question is, so both assertion and reason are correct, and reason is the correct. Sorry, both assertion and reason are correct, and reason is the correct explanation of the particular assertion. And the option B states that both assertion and reason are correct, but reason is not the correct explanation of the assertion. And the option C says that assertion is true, but reason is false. And the last option is both assertion and reason is false. So anyone would like to join? Hmm? Anyone? Okay, let me do this first.
Um,
Please
so
continue.
assertion is true, reason is false actually.
Okay, so which option?
Sir, option C.
Option C. Assertion is true, but reason is
Um, s-
false. So any reason behind this? Any like
sir, actually,
supportive statements?
sir, actually, using the restriction enzyme, after introducing it in survive in penicillin mixed plate, just survived in streptomycin added plate actually. That means it's able to survive in streptomycin added plate. That means like it won't be that sensitive to streptomycin added plate.
Mhm.
So it will be the other way around.
Okay, correct. So let me explain this. So we'll start from the streptomycin resistant gene. So in molecular biology, an antibiotic resistant gene serves as a crucial marker during genetic engineers and others. So typically incorporated into vectors such as plasmids. So this gene confers resistance to a specific antibiotic. So when introducing a foreign gene, wait. Huh. So when introducing a foreign gene into a host organism, so the vector along with the antibiotic resistant gene and the desired DNA fragment is introduced. So the subsequent plating of the transformed cells on a growth medium containing the corresponding antibiotic creates selective pressures. So only the successfully transformed cells with have the incorporated the vectors and consequently the foreign DNA. So here in this scenario, in the streptomycin resistant gene and in 3, so here it's cut site located in the penicillin resistant gene. So after the insertion of the gene of interest, so bacteria are plated on two different types of plates. So one containing the penicillin and other containing the streptomycin. So the rationale of these statements are the hint 3 digestion. So here the hint 3 digestion cuts the vector at its recognition site. So which happens to the within the penicillin resistant gene, that this cleavages creates a gap in the penicillin resistant gene. And the second supports for this is insertion of gene of interest. So here the gene of interest is then inserted into the gap which was created by Hindtree digestion. So this insertion disrupts the penicillin resistant gene. And the last and third statement which supports this assertion is the plating on penicillin and streptomycin plates. So here the bacteria transformed with the vector containing the disrupted penicillin resistant gene will not survive on plates containing penicillin. So because the gene is no longer functional, however, so they will survive on plates containing streptomycin due to the presence of the vector streptomycin resistant gene. So based on this supportive information, so assertion is true but reason is false. So here one term is used transformants. So I've told you in the question 4 during gene cloning what transformants is. So transformants come from the term transformation. So the recombinant DNA which was introduced into host organism, usually a bacterial cell, through a process called transformation. And then comes to the selection of transformants. So to identify cells that have successfully taken up the recombinant DNA, a selectable marker like such as antibiotic resistance gene, so which is often included in the vector. So only cells that successfully incorporate the gene of interest will survive the presence of the selective agents. Okay, so
Yes, I got it.
option C, which states the assertion is true but reason is false, is the correct answer. So moving to the last question for today. So is the one of the popular methods of transformation of recombinant plasmids to host cells is to perform heat shock transformation. So what can be the best explained as the role of calcium chloride during heat shock transformation? So anyone would like to explain what heat shock transformation is and what is the role of calcium chloride during the heat shock transformation? Mhm.
Sir, actually, calcium chloride, what it does is it just makes the cell membrane permeable, actually.
Exactly.
So it is
Exactly.
option B.
Go ahead, go ahead.
So, in fact, when it makes the membrane permeable, then for some amount of time, like the bacteria, it goes into a shock, kind of like shock. So, which is called as heat shock, actually. And because of that, then our gene of plasmids containing a gene of interest can easily enter that cell.
Okay. So, according to you, what is the best explained option here?
Sir, option B.
Option D, B or D?
Sir, B.
Okay. Which states that it makes the cell permeable, membrane permeable to accept foreign particles. Okay. So, the option C is the right one.
Acha, okay, sir. Got it.
Why? Why option C is the correct?
Sir, mostly because it just changes the charge
Charges,
orientation of
exactly. It
the cell
changes the charge
membrane.
in the bacterial cell wall, changes the actually bacterial cell wall is negatively charged and DNA cell membrane is negatively charged. So both are same charge. So both same charges will repel each other. Okay. So what
Okay,
you
sir.
are saying is
Got it.
option B, which is make permeable to accept foreign particles. So foreign particles, like there are a lot of foreign particles. So anyone could invade into the cell membrane. So we have to be specific at that time. So which is the mechanism by which it makes the positive charge to the cell wall. So let me explain this. So with an example, with a diagram. So this is the diagram. As you can see, DNA and the bacteria. Hmm? So here in heat shock transformation, a widely used method for introducing recombinant plasmid into host cells, calcium chloride plays a crucial role in enhancing the efficiency of the process. So the presence of calcium chloride serves multiple functions during heat shock transformation. So firstly, calcium chloride helps neutralize the negative charges on the DNA and bacterial cell membrane. Both are negatively charged. DNA also negatively charged and the bacterial cell membrane is also negatively charged. So followed by, so firstly it will neutralize the both the charges. So then facilitating the binding of DNA to the cell surface. So after this, neutralization is crucial for promoting electrostatic interaction between the negatively charged DNA and the negatively charged bacterial cell membrane, which allows the plasmid DNA to adhere or attach to the cell surface. And followed by the secondly, calcium chloride is involved in the formation of small pores or transient disruptions in the bacterial cell membrane, which is a positively charged cell membrane. So these disruptions enable the uptake of the plasmid during the heat shock phase. So it here calcium chloride act as a carrier. So where is a brief exposure to the cell to elevated the temperature enhances the membrane fluidity and permeability by making it to the positively charged cell membrane. So consequently, the combination of calcium chloride treatment and heat shock increases the efficiency of plasmid uptake by the bacterial cell, leading to the successful transformation. So this, according to this explanation, the option C is the right one, which states that it gives the positive charge to the cell wall. Okay,
Yes, I got it.
so we're done for the day. So we'll meet in the next session, next Tuesday. Okay,
Okay, sir.
okay. Chalo, thank you. You guys can leave the meeting. Bye bye.
Thank you, sir.
Thank you, sir.
Thank you, sir. Bye.
Thank you. Good night to
Yes,
all.
sir. Thank you, sir.